The dream of a lab on a chip is as simple as it is revolutionary. Shrink an entire laboratory's worth of diagnostic tools onto a piece of glass or polymer smaller than a credit card. Instead of mailing blood or saliva to a distant lab and waiting days for results, a chip the size of your fingertip could separate cells, detect pathogens, measure biomolecules, and deliver answers in minutes. Doesn't that sound like carrying a hospital in your pocket? What part of delivering a lab cannot be automated and deliver answers in minutes? Doesn't that sound like carrying a hospital in your pocket? The power comes from microfluidics, the art of steering tiny volumes of liquid through channels thinner than a hair. At this scale, fluids flow smoothly and predictably, allowing exquisite control. Droplets can be split, mixed, or merged like logic gates. Antibodies or DNA probes can be tethered to channel walls. Sensors can read out fluorescence, electrical signals, or even mechanical changes. A lab on a chip becomes not just a miniature lab, but a programmable diagnostic platform. The real promise lies in accessibility and speed. Imagine a rural clinic without advanced infrastructure being able to test for malaria, HIV, or tuberculosis on the spot. Or consider cancer diagnostics. Circulating tumor cells in a blood sample are extraordinarily rare, yet a microfluidic chip can filter and capture them with a precision impossible for bulk methods. In emergencies, chips could diagnose infections or organ failure within minutes, fast enough to save lives. A lesser-known fact is that many chips also aim to be self-contained ecosystems. Some designs use capillary action, not pumps, to move fluids, eliminating the need for bulky equipment. Others embed reagents in dried form, activated only when a sample enters. Some even integrate smartphone cameras for optical readout, turning ordinary phones into portable laboratories. Isn't it remarkable that the same device used for selfies could one day also diagnose pneumonia? Yet challenges remain. Biological fluids are messy, clogging tiny channels. Manufacturing chips cheaply and reliably is not trivial, and perhaps the deepest paradox is this: while chips are designed for simplicity, their internal physics, mixing surface effects, evaporation, are anything but simple. Engineers must embrace noise, variability, and instability to make diagnostics robust outside pristine labs. The haunting insight is that lab on a chip is not just a miniaturized laboratory, it is a new philosophy of diagnostics. Instead of centralization, large labs processing thousands of samples, it decentralizes testing, placing power directly in patients' hands. If successful, it could transform medicine from reactive to preventive, catching disease at its earliest whispers. And perhaps the greatest revolution will be psychological. To make the invisible processes of our bodies visible instantly. On a chip, lab-on-a-chip devices condense full laboratory workflows into a few square centimeters of microfabricated channels, handling fluids in picoliter or nanoliter volumes and drastically reducing reagent use and cost. At these scales, surface forces dominate, so liquid behavior is controlled with precision using geometry and chemistry. Pumps, valves, mixers, and detectors can all be embedded into a single platform, integrating processes that once required bulky instruments, which enables decentralized, portable, and efficient testing directly at the point of need. A chip that measures blood glucose from a single droplet without requiring a full laboratory setup shows this potential clearly, like shrinking an entire kitchen into a handheld Swiss Army knife. The main value of LOC lies in its ability to deliver results at the point of care rather than at distant facilities, allowing patients to benefit from real-time information and enabling immediate clinical decisions. This reduces delays associated with sample transport, centralized analysis, and result reporting, while also making diagnostics possible with minimal training. Their portability means they can be deployed in rural clinics, emergencies, or epidemics, cutting logistical barriers and democratizing access to healthcare. Portable ALOC devices for HIV detection that provide same-day results in remote regions are an example, much like having a pocket doctor that delivers answers instantly rather than waiting days. LOC platforms combine diverse diagnostic steps into a single enclosed environment, allowing cell lysis, nucleic acid amplification, protein detection, and signal readout to occur seamlessly in sequence. This integration reduces error, since human handling is minimized and cross-contamination risks drop, while also increasing speed by eliminating transfer delays. Customization enables disease-specific chips with embedded reagents, creating self-contained diagnostic tools requiring little external equipment. A cartridge-based low system that automatically processes a nasal swab to detect SARS-CoV-2 exemplifies this, operating like a vending machine that performs the entire recipe automatically, from ingredients to finished meal. Miniaturization also concentrates analytes into tiny volumes, effectively amplifying signals without additional reagents. High surface-to-volume ratios allow rapid interactions between molecules, while nanostructures or engineered surfaces selectively bind targets with remarkable fidelity. This makes it possible to detect rare biomarkers, such as single cancer cells or DNA mutations, with improved precision due to reduced background noise at the microscale. Digital droplet PCR chips that detect a single mutated DNA strand among thousands of healthy ones illustrate this capability, much like a fine-tuned microphone that can hear a whisper in a crowded stadium. The AM factor, the automation further shortens diagnostic timelines as LOC systems control fluid motion with capillary forces, electric fields, or micropumps rather than manual pipetting. This ensures consistency across samples, minimizes human error, and provides rapid, high-throughput results. Speed is especially critical in emergencies where treatment decisions must be made immediately. For example, cardiac biomarker chips can deliver results for heart attack screening in under 20 minutes, replacing a long relay race with a high-speed conveyor belt. Yet, despite impressive prototypes, many LOC devices struggle to transition from laboratory demonstration to clinical practice. Manufacturing at scale requires standardization and cost reduction, while reagent stability outside controlled labs poses practical issues. Devices must also withstand variability in real-world samples, environments, and users, while passing rigorous regulatory approval processes. Without solving these problems, many LOC systems remain confined to academic research. The fate of numerous cancer detection chips that never reached hospitals resembles concept cars that never make it onto everyday roads. Still, the implications for global health are transformative. LOC devices can be designed to operate without electricity, refrigeration, or complex instrumentation, using inexpensive substrates such as paper or polymers. They provide affordable, disposable tools that empower healthcare workers in resource-limited settings, enabling on-site detection of diseases like malaria or tuberculosis. Paper-based LOC tests that detect malaria parasites from a finger prick of blood exemplify this accessibility, much like giving every village its own miniature power station for health. The technology also increasingly connects with digital ecosystems. Smartphone cameras can capture assay results. AI algorithms can analyze subtle patterns invisible to the human eye, and cloud networks can aggregate data for epidemic monitoring. This convergence of biology, computation, and connectivity turns every test into both a personal diagnosis and a public health data point. AI-enhanced ELOC platforms that mapped COVID-19 outbreaks by aggregating results across communities demonstrate the power of this synergy, like transforming each test into both a personal medical answer and a node in a global radar system. Looking ahead, LOC will not replace centralized laboratories but will complement them with unique capabilities. Its strength lies in decentralization, ultra-low volumes, and digital integration, with future designs potentially including wearable chips embedded in patches or clothing. Multiplexing may allow simultaneous detection of dozens of biomarkers, and self-ported chips could make diagnostics autonomous. Wearable sweat-analyzing chips that track hydration and electrolytes during exercise foreshadow this future, much like carrying a personal weather forecast for your body at all times.